My name's Abby Jones, and I'm a sophomore biochemistry major. I work with Dan Panaccione in his lab, and our project is on the discovery of pharmaceutically important ergodalkaloids found in 3 species of the fungal genus Aspergillus. Ergodalkaloids are a family of specialized fungal metabolites that contaminate grains and grasses in agriculture, but can be modified into useful pharmaceuticals. They have been prescribed in drugs to treat Parkinson's disease, migraines, uterine hemorrhaging, and a variety of other conditions. Now, there are 2 major types of ergodalkaloids: lysergic acid derivatives, which are used for the production of these important pharmaceuticals, and then clavines, which have not been found yet to be useful. Similarly, there are 2 major groups of fungi known to produce ergodalkaloids: plant-associated Clavicipitaceae and the molds of Aspergillus and Penicillium species. The Clavicipitaceae species produce lysergic acid derivatives, which are beneficial for pharmaceutical production, and the Aspergillus and Penicillium species produce different, less desirable ergodalkaloids. The fungi in the Clavicipitaceae don't grow well in culture, however, and they're difficult to work with genetically, making them less useful industrially. The Aspergillus species, on the other hand, are fast-growing and very easy to culture and manipulate genetically, making them the ideal species to work with industrially. However, prior to our work, there have been no species of Aspergillus known to produce lysergic acid derivatives. If we could find species of Aspergillus that could make lysergic acid derivatives, they would be an optimal group of fungi to work with within the production of pharmaceuticals. We hypothesized that there are Aspergillus species that are capable of producing lysergic acid derivative ergodalkaloids. To test this hypothesis, we used a genome mining approach. To start, we genome mined the whole genome shotgun sequence database of Aspergillus species. We T-blasted these sequences looking for DMAW and EZA, which are 2 genes that are necessary for the production of ergodalkaloids. We acquired cultures of these fungi and then tested them using high-performance liquid chromatography, the HPLC, and mass spectrometry. We used high-performance liquid chromatography to determine the identity of the chemicals being produced by the fungi. We discovered the accumulation of stereoisomers of lysergic acid alpha-hydroxyethylamide, also known as LAH. As you can see in the graph below, Aspergillus homomorphus produces an extremely large amount of this chemical, far exceeding the amount present in Metarrhizine broneum, a fungi known to produce high amounts of LAH previously. Aspergillus leprous and Aspergillus hancockii, which is not shown on the graph, also produce large quantities of LAH. Additionally, farther testing using mass spectrometry also supports the identity of LAH. We can tell this because the mass and retention time of our sample was identical to that of LAH and Metarrhizine broneum that we had previously confirmed. Aspergillus leprous produces large quantities of LAH, but over time, most of this LAH is hydrolyzed to ergine, which is another lysergic acid amide, and then eventually degraded. Aspergillus homomorphus, on the other hand, which accumulated 200-fold higher concentrations, maintain these high concentrations for at least 50 days, which is ideal, compared to Aspergillus leprous, which begins hydrolyzing to ergine after about 9 days. Still, both LAH and ergine could be a useful scaffold for synthesizing lysergic acid-derived pharmaceuticals. Since Aspergillus leprous is the species that we are able to acquire first, we've conducted some additional experiments with this species. We tested Aspergillus leprous for the secretion of ergodalkaloids into the liquid medium of the fungus. We did this by separating the solid hyphae and the liquid medium of the fungal culture. We found that in Aspergillus leprous, most of the ergodalkaloids are secreted into the liquid. This characteristic is significant because the fungus's ability to secrete ergodalkaloids is beneficial for purification for pharmaceutical purposes. Additionally, preliminary data shows that Aspergillus homomorphus also is doing the same thing, with secreting most of the ergodalkaloids into the liquid medium of the culture. Since the types of ergodalkaloids produced by Aspergillus leprous, Aspergillus homomorphus, and Aspergillus hancockii are similar to the alkaloids produced by Metarrhizine broneum, which is a known insect pathogen, we tested their ability to infect insects. We did this by injecting 3,000 spores of each fungus into the larvae. As shown in the kill curve below, Aspergillus leprous and Aspergillus hancockii infected the larvae and killed it. Additionally, following the insect's death, the fungi sporulated on the insect, and high concentrations of ergodalkaloids were present on the insect after its death. we confirmed this by using the HPLC. We also tested the fungus's ability to naturally infect the larvae by placing them on a plate of fungal culture. Once again, Aspergillus leprous and Aspergillus hancockii infected the insect and emerged on them after their death, indicating that they are also natural insect pathogens. The 4 photos below show sporulation on the insect after their death. The 2 photos on the left are infected by Aspergillus leprous, and the 2 photos on the right are infected by Aspergillus hancockii. The species we used to test all these was Galleria melanella. We chose Galleria melanella because it has been frequently used as a model of pathogenesis in mammals. Therefore, our results indicate that Aspergillus leprous and Aspergillus hancockii have the potential to be mammal pathogens, though there is no known cases of pathogenesis by these species reported yet. To further test the fungi's ability to be a mammal pathogen, we grew Aspergillus leprous at 3 different temperatures: 20 degrees, 30 degrees, and 37 degrees. We did this because in order for it to be able to infect mammals, it would need to be able to grow at 37 degrees, due to having to overcome a mammal's body temperature. The fungus was capable of growing at all 3 temperatures, but it only produced ergot alkaloids at 20 degrees Celsius and 30 degrees. This shows us that if ergot alkaloids are necessary for the infection of mammals, then the host range would have to be limited to species with a lower body temperature. Another study we did was focused around new genes found in Aspergillus leprous. Aspergillus leprous contains many of the same genes as other ergot alkaloid producing fungi, but it has 2 previously uncharacterized genes, one of which we called EAST, which is unique to Aspergillus leprous. This gene has sequence properties of a major facilitator transporter. Due to its location among ergot alkaloid associated genes, we hypothesized that it was involved in ergot alkaloid production or secretion. To test this hypothesis, we expressed this gene into a strain of Aspergillus fumigatus that was previously engineered to produce the important pharmaceutical compound lysergic acid, but it only produced it at poor yields. To express EAST in the strain of Aspergillus fumigatus, we joined the coding sequence of EAST from Aspergillus leprous with an Aspergillus fumigatus promoter, as shown below, and then we transformed it into the fungus. We used Aspergillus fumigatus strain SR17 as the transformation recipient because it had been engineered previously to produce lysergic acid, but it had only produced it in small amounts, indicating that the artificially engineered pathway might be improved upon. Transformed colonies were screened by PCR to identify that those colonies had taken up the modified EAST gene. Overall, more ergot alkaloids were produced in the EAST transformed fungus, and most of them were retained within the fungal hyphae. Instead of producing lysergic acid like the parent strain SR17, the EAST engineered fungus accumulated higher levels of agroclavine, which is lysergic acid's immediate precursor. Additionally, there were high concentrations of chanooclavine, which is agroclavine's immediate precursor. So since the sequence of EAST indicates that it acts as a transporter, our results suggest that it is being used to transport ergot alkaloid intermediates within the cell to different compartments, as opposed to our previous theory of them transporting ergot alkaloids in and out of the cell. Retention of these intermediates within the cell allows for more ergot alkaloids overall to be produced than in the parent strain, but this presumed compartmentalization prevented the eventual production of lysergic acid. Overall, we have 4 major discoveries in our project. First, we found that at least 3 Aspergillus species are capable of producing lysergic acid derivative ergot alkaloids. This will be incredibly useful in the production of pharmaceuticals in the future. Next, we found that Aspergillus leprous secretes most of its ergot alkaloids into the liquid medium of the culture, which again is beneficial for purification and incredibly useful industrially. Third, we found that Aspergillus leprous and Aspergillus hancockii are capable of infecting insects. And fourth, we found the novel gene EAST increases the production of ergot alkaloids within the fungus. And additionally, there is possibly a role that EAST plays in the transportation of these ergot alkaloids intermediates within the fungus. This work was facilitated by Shai Seen, who helped a lot with insect projects, Sammy Fabian, who taught me basic lab techniques, and it was funded by an NIH grant.